Hello. In this video, we will discuss about the drug actions, the mechanisms of the drug actions. So, the action of the drugs on the organism is called as the pharmacodynamics. The drugs produce their effects by interaction with the discrete target biomolecules. Generally, these biomolecules are proteins. The functional proteins that are targets of the drug action can be grouped into four major categories. So, enzymes, ion channels, transporters, or the receptors are the functional proteins that are targets of drug action. So, we'll see one by one what are the different enzymes, ion channels, and transporters and receptors, how they act as the targets for the drugs. What are the enzymes? Enzymes are nothing but the proteins. They are biological catalysts. They can change the rate of chemical reaction without needing an external energy source. So, almost all biological reactions are carried out under the catalytic influence of the enzymes. Hence, these enzymes are very important targets of the drug action. So, drugs can either increase or decrease the rate of enzymatically mediated reactions. However, in physiological systems, enzyme activities are often optimally set. Thus, stimulation of the enzymes by drugs that are fully foreign substances is unusual. So, enzyme stimulation is relevant to some natural metabolites only. For example, pyridoxine acts as a cofactor for the increase in decarboxylase activity. So, several enzymes are stimulated through the receptors and secondary messengers also. For example, adrenaline stimulates the hepatic glycogen phosphorylase through the beta receptors and cyclic AMP. So, the stimulation of enzyme increases its affinity to the substrate so that rate of constant, that is the Km value of the reaction, is lowered. So, stimulation of enzyme increases the affinity for the substrate and Km value will be reduced. So, apparent increase in the enzyme activity can also occur by the enzyme induction. So, enzyme induction can increase the enzyme activity. So, that is synthesis of more enzyme protein. That cannot be called as a stimulation because the Km does not change here. So, rate constant is not changing. But here you can see the synthesis of more protein. Inhibition of enzymes is a common mode of drug action. So, which is the common mode of drug action? Enzyme inhibition. So, here two types of inhibition you can see that non-specific inhibition means many chemicals and drugs are capable of denaturing the proteins. They alter the tertiary structure of the enzyme with which they come in contact and then it inhibits. So, inhibition of enzyme is the drug action. So, heavy metal salts, strong acids, alkalis, alcohol, formaldehyde, phenol inhibit the enzymes non-specifically. Non-specifically means they just alter the tertiary structure and then they can come contact with this one. So, such inhibitors are too damaging to be used systemically. So, because they are changing the tertiary structure of the enzyme itself. So, specific inhibition, if you see, many drugs inhibit a particular enzyme without affecting the others. Such inhibitors are either competitive inhibitors or non-competitive inhibitors. So, competitive inhibitors are again you can divide them into equilibrium type or non-equilibrium type. So, competitive inhibitors in equilibrium type, you can see the drug is being structurally similar and competes with the normal substrate for the catalytic binding site of the enzyme so that the product is not formed or the non-functional product is formed. and a new equilibrium is achieved in the presence of the drug. Such inhibitors increase the Km value, but the Vmax remains unchanged. So higher concentration of the substrate is required to achieve the maximal reaction velocity. So, but if the substrate concentration is sufficiently increased, it can displace the inhibitor and the same maximal reaction velocity can be seen. So that means in competitive equilibrium type, Km value increases and Vmax value remains unchanged. So the examples for this competitive inhibition is neostigmine competes with the acetylcholine for the choline esterase, or the other example is captopril competes with the angiotensin 1 for angiotensin converting enzyme. So coming to the non-equilibrium type of enzyme inhibition, this occurs with the drugs which reacts with the same catalytic site of the enzyme, but either forming the strong covalent bond or it has such a high affinity for the enzyme so that the normal substrate is not able to displace the inhibitor. The best example for this is methotrexate has 50,000 times higher affinity for the dihydrofolate reductase than the normal substrate DHFA. So that the clinicians, whenever they prescribe the methotrexate to the patients, they also prescribe the high doses of folic acid in the clinical practice. So in these situations, Km is increased but Vmax is reduced. Whereas in the earlier situation, Km is increased, Vmax remain unchanged. Coming to the non-competitive inhibitors, so these inhibitors reacts with the adjacent site and not with the catalytic site. But it could be able to alter the enzyme in such a way that it loses the catalytic property. So hence the Km is unchanged but Vmax is reduced. The examples are acetozolamide competes with the carbonic anhydrase, and disulfiram competes with the aldehyde dehydrogenase, and omeprazole it competes with the H plus clay plus ATPase. So the disulfiram is a chemical substance which is used for the de-addiction. So whenever the disulfiram is given to the patient, so the aldehyde levels will be increased and the patient will have the aversion for that this one. So it is a non-competitive inhibitor. Now we will see the ion channels. Proteins which acts on ion selective channels participate in the transmembrane signaling and regulate the intracellular ionic composition. So this makes them a common target for the drug action. So the drug can affect ion channels either through the specific receptors, or specific receptors means ligand gated ion channels or the G protein operated ion channels, or by directly binding to the channel and affecting ion movement through it. Local anesthetics which physically obstruct the voltage sensitive Na plus channels. In addition, certain drugs modulate the opening and closing of the ion channels. Example, quinidine. Quinidine blocks myocardial Na plus channels. Nifedipine blocks L-type voltage sensitive Ca plus 2 channels. Sulfonylureas, these are the drugs, hypoglycemic agents. These will inhibit the pancreatic ATPase sensitive K plus channels. So the next one is amiloride. Amiloride inhibits the renal epithelial sodium channels. This picture shows about the ion channels. So next one is the transporters. Several substrates are translocated across the membranes by binding to the specific transporters. So there are specific transporters or the carriers. So these are also called as a carrier, which either facilitate the diffusion in the direction of the concentration gradient or pump the metabolite against the concentration gradient using the metabolic energy. So active transport and passive transport I am talking about. Many drugs produce their action by directly interacting with the solute carriers to inhibit the ongoing physiological transport of metabolites. So here the solute carriers I am using. Solute carriers means these are the class of transporter proteins. So best example for this one, the dopamine transporter, vesicular amine transporter, GABA transporter. Amphetamines selectively blocks the dopamine reuptake in the brain neurons by dopamine transporter. Reserpine blocks the granular uptake of noradrenaline and 5-HT by vesicular amine transporter. The anticonvulsant tiagabines act by inhibiting the reuptake of GABA into brain neurons by GABA transporter, that is GAT1. So majority of the drugs do not bind directly to the effectors such as enzymes, channels, transporters and structural proteins and template biomolecules. But they act on the specific regulatory molecules that control these effectors such as channels, transporters, structural proteins, templates and other biomolecules. These regulatory macromolecules are the sites on these regulatory molecules which bind and interact with the drugs are called as the receptors. That means receptor means, so receptors definition, if you see that it is a macromolecule or a binding site located on the surface or inside the effector cell that serves to recognize the signal molecule or the drug and initiate the response to it, but itself has no other function. So that inactive receptor will be there. Once the ligand comes and binds to that inactive receptor, that receptor will be activated, and you can see that all post receptor events are the signaling cascade you can see in the cell effectors. So what are the ligands? Now ligand means in the Latin word ligare, it has to bind. Any molecule which attaches selectively to a particular receptor or the site is called as a ligand. So the term only indicates the affinity or binding without regard to the functional change. Agonist and competitive antagonists are both ligands of the same receptor. See, any receptor will have some agonists or some competitive antagonists. If the agonist, full agonist bind to this one and the strong effect can be seen, if the partial agonist can attach to the receptor, this is receptor and this is the ligand. This is receptor and this is the ligand. Here this is the receptor and you can see these ligands. So if some partial agonists are coming and binding to the receptor or receptor binding site, then you can see that little effect. And if the antagonist bind to this receptor, and you can see that no effect will be seen. Pharmacologic antagonist, if you see this, antagonist is an agent that activates the receptor to produce an effect similar to the physiological signal molecule. So the inverse agonist is an agent that activates a receptor to produce an effect in the opposite direction to that of the agonist. So this here agonist is showing here full agonist or partial agonist is showing that positive response, and whereas the inverse agonist is showing the negative response. Opposite direction you can see the effect. So partial agonist means an agent that activates the receptor to produce the submaximal effect but antagonizes the action of the full agonist. Coming to the antagonist, an agent that prevents the action of an agonist on a receptor or a subsequent response but does not have any effect on its own. The other characteristics of the receptors are selectivity, affinity, and efficacy. Selectivity means binding of the drug to a receptor which depends on the physicochemical structure. And second one, affinity means the strength of the binding between the drug and receptor. And third one, efficacy or intrinsic activity means the ability of the drugs to elicit the pharmacological response after its interaction with the proper receptor. Thank you very much for the patient listening. If you like, you can share this video to other